all the audience from IIT Madras. Welcome to the Micro Nano Biofluidics monthly talk series. We are indeed delighted and honored to have Professor Stone from Princeton as a speaker for today's talk. Thank you, Professor Stone, for accepting our invitation. As many of you already know, Professor Stone is a pioneer and one of the topmost researchers in the field of fluid dynamics. His research is focused at the interface of engineering, chemistry, physics, and biology, and which has indeed pushed the frontiers in the areas of microfluidics, multiphase flows, electrokinetics, flows involving active matter, biofilms, and so on. He has received several accolades and honors, including the GK Bachelor Prize in Fluid Dynamics, APS Fluid Dynamics Prize, and many more.
It's okay, Professor Sen.
Yeah, thank you. And thank you, Professor Stone, for accepting our invitation. And today's talk will be on microfluidics approaches to new problems, separations, diffusiophoresis, and soil carbon. Professor Stone, over to you.
Thank you, Professor Sen. Can you see my screen again?
Yes, we can, and we can also hear you clearly.
Okay, perfect. So I will just start. So thank you again for the invitation. I placed on the front the Department of Mechanical Engineering, and Professor Sen had shared with me the idea that many of the people here will be interested in microfluidic ideas. So you may be aware my group is not the most technologically focused group, but I picked three themes that we've worked on. The first one a few years ago, and then the last two more recent, that discuss how we've used microfluidic ideas to look at an idea for separations, a chemically driven transport process called diffusiophoresis, and a question that some colleagues and I discussed that seemed very important and interesting on the issue of soil carbon as it relates to climate modification. So I have to thank my research group. They probably have most, if not all, of the ideas in this talk, and this picture was taken just before the pandemic hit almost 20 months ago now. Okay, so I'm going to just start with a little overview of just different things I get to think about. I called this a few different problems in fluid mechanics, but I picked sort of the microfluidic side of things, except for one example. I'm going to tell you about a fluid mechanics problem that I really like called the Bretherton problem, and how it comes up in motions of bubbles, and how we turn that into a question about filtration. I'm going to guess a few people, though maybe not many on the call, know about electrokinetics, and there's a part of it called diffusiophoresis, which says what the words mean. Phoresis means the movement of particles. Diffusion means there's a chemical gradient, and so this is how chemical gradients move things. And finally, if there's time at the end, this issue of carbon, soil carbon, and carbon storage, and questions that come up in climate questions that we tried to approach with microfluidics. So far, so good? Is anybody there?
Oh, yes, yes.
Yeah, okay. Yeah, I tend to get nervous that everyone has gone away and I'm all alone. Okay, so this first thing I want you to think about, these are just a set of examples in fluid mechanics, is a bacteria called Pseudomonas Originosa. It's about a micron in dimensions. It has the ability to move on surfaces, not by using a flagella. In fact, it loses its flagella, but it has a set of actuated polymeric structures called pili, and when it moves them, it can pull itself on surfaces. And generally speaking, this surface motility had been thought of as a sort of random motion. But a few years ago, a number of years ago now, probably more than I want to think about, we made the following observation. There's a microfluidic device in the upper left that creates flow from one side to the other. In the movie, the flow is going to go from the top to the bottom of the page. The small black dots you see are the bacteria, Pseudomonas Originosa. They're each about a micron in length. This bacterium will kill you if you have a compromised immune system. And when you start the flow, you might expect and this is what the literature would say, that the flow creates a stress and the stress should move everything from top to bottom. In fact, this is also what every mathematical model of these kinds of problems state. But if you do the experiment and I ask the audience, which way are the bacteria moving? Well, assuming the movie's playing well for you, which way do you see the bacteria moving?
Movie is playing well. I think maybe you have to play it again.
Yeah, so the flow is top to bottom, but which way do you see the bacteria migrating?
In the opposite direction.
Yeah, in the opposite direction. So these bacteria migrate upstream. I think we were probably the second research group to notice this. And the feature of it is it's an interplay between the bacterial orientation, which is in some sense set by the flow, and the surface motility that I showed you on the previous slide. And the sketch in the upper right of this slide shows that idea that the bacteria, when they're able to attach to the surface due to polar localized pili, should get oriented by the shear flow near the surface. And then when these pili are actuated and they pull, the bacteria move upstream. So that was a surprise we discovered in microfluidic experiments that we then studied much more carefully. Another thing we studied was a feature of the shape of curved cells. This is joint work also with Zamar Gittai in molecular biology at Princeton. The observation is that some cells have a natural curvature. That's shown here. If you look at the sketch on the bottom left, the cells have a natural curvature. And at the time of our work, it was unclear what the benefit was of being curved. If you looked at the simple growth of bacteria, a curved cell and a mutant that was straight grew at the same rate. And what we discovered using a microfluidic experiment is that the benefit of the curvature of the cell meant that when you try to multiply your number, which required cell doubling and then reattaching to surfaces to colonize the surface, that being curved meant you were more likely to colonize surfaces. And we did experiments both with wild type and mutant bacteria and showed that the advantage of the curved cell was that, and the image on the right has green being the curved cell, means you're more likely to colonize the surface. So we were able to test a hypothesis of the role of flow of providing a benefit to a cell, in this case, a shape of a cell, which hadn't been identified before. So that was something we did with microfluidics in collaboration, again, with people in molecular biology. Finally, I'll leave you with this one. Quorum sensing is a way that multi-cell communities can communicate. They communicate by the cells excreting molecules that are counted by the community. And it's known in many cases that boundaries can be rough. The image on the left is an organ inside you that has kind of a folded structure. You know, if you get cavities in teeth, the surface can be rough. And there are many rough surfaces or pipes in engineering. And we were interested in bacterial colonization of the surfaces and the fact that they might quorum sense. And I'll just show in the movie on the left that you can do microfluidic experiments that pretty much show that when the flow goes over a rough surface, very little of the flow can penetrate the roughness, the characteristic features of the undulations of the surface. And if you're probably all experts on low Reynolds number flows, and you can appreciate then the differences between the external flow and the flow in the roughness. If you then do an experiment with bacteria that can quorum sense, which means if their number gets sufficiently large, they can undergo a biochemical transition. You do a flow experiment. There are bacteria everywhere multiplying, those you show by black, but the flow sweeps away quorum sensing molecules. And so this movie shows that the only quorum sensing, which here is indicated by the green fluorescence, The only quorum sensing possible is in the area without flow, which is the texture, not in the same spatial part above the place. And so this was one of the early demonstrations that said how genes get expressed by bacteria can also be influenced by flow, which means you not only get spatial variations, but temporal variations, and they're actively linked together to the flow in the environment. And again, this was work with Bonnie Bassler's group in molecular biology, again, using microfluidic techniques. Finally, I'll just leave you something fun for those of you who like fluid mechanics. I received an email some years ago, shown in the upper right, asking about layers that form in a cup of hot latte. The images on the left are a time snapshot when you pour coffee into hot milk. It's important to be hot. It has to cool to the surroundings. As you watch this system, you might see on your screen, if the image comes through well, that in the image labeled t = 2 minutes, there are horizontal strata in the coffee. If I play a movie of this, you'll see it effectively in a sped up movie that first starts at real time and then is sped up a bit. There's a fluid mechanical phenomena that involves both horizontal temperature gradients and vertical concentration gradients due to pouring of the coffee into the hot milk. And the layering appears driven by the fluid flows that are density driven by these density gradients. And since there are two of them, this is actually a mimic of double diffusive convection, which is well studied in oceanography, here studied in a completely uncontrolled way, but at the countertop because of a beverage you might like to drink. And we studied that problem, though I forgot to put the reference on the slide. Okay, so those are my brief introductions of fluid mechanics problems. And now I want to tell you about 3 short stories that involve microfluidic approaches to different problems. And the first one I want to tell you about is separations idea. So separating particles from one another, say by size or some other attribute, and a fluid mechanics problem that I think is one of the great fluid mechanics problems. And I love to tell people about it. Its abbreviation is called the Landau-Levitch-Darjagan-Bretherton problem, sometimes called the LLDB problem. And I'll show you this, and this is joint work with my former graduate student, Estella Yu, and a postdoc, Seppa Kotoparas, who's now in the faculty at the University of Leeds. So there are many different processes that allow you to do separations in industry, whether it's centrifugation or membrane filtration, or a very classical idea that it can come up in many different ways called field flow fractionation, or flow field fractionation, I guess. And I put a number of sketches on the right. Several of these are microfluidic, modern versions of microfluidic methods to use these ideas. And the feature of all of them is that they try to implement in a controlled way, in a laminar flow, these kind of separation processes. I won't go through them with you in the interest of time, but happy to talk to you about them. But suffice it to say, there are a large number of separation processes, and there's a large literature in microfluidics, people trying to apply and or invent new separation processes, or just test the basic underlying scientific principles. Okay, so that was separations. How about fluid mechanics? So here's my favorite fluid mechanics problem. You take a flat plate indicated on the slide, you pull it at speed capital U through a liquid that has density rho and viscosity mu, and you want to predict the film coating denoted H that wets the surface as the plate is withdrawn. Surface tension gamma acts at the interface and gravity acts down. So the question is, what's the formula for H? Can you determine that? And that was first worked out 80 years ago by Landau and Levitch. And a related problem involves not a plate, but a cylinder of radius B that's pulled from the fluid again at a certain speed. And the question is, predict the thickness H. That was sorted out by Boris Deryagan, who's arguably the greatest physical chemist of the 20th century. Okay, so these are two different problems. So you might not be so impressed, but I'm going to show you three more. If a bubble moves in a channel or in a tube, if you turn your head on the side, you see a thin film connected to a curved meniscus. So this, if you turn your head on the side, this looks just like the Landau-Levitch problem. If you do any kind of coating flow process, in this case, a roll coating process where a cylinder is dipped in a fluid and rotated, it drags up a thin film. Then you have a thin film where I'm pointing of thickness that you'd like to predict H connected to a curved meniscus. And finally, if you want to coat the inside of a cylinder or some kind of light bulb or some other surface, you place the liquid there and you might spin the boundary. And again, you get a film coating process. So these are 5 different problems. I'll just maybe flip back again. 2 here and 3 here, 5 different problems, and they all have the same solution, which when I stumbled on, I didn't invent this, but I stumbled on it. I found it sort of incredible. And the universal solution says that the film thickness relative to the length scale characteristic of the meniscus curvature, here denoted L meniscus, and that varies from problem to problem, is some constant times the capillary number to the 2/3, where the capillary number is the viscosity, the substrate speed, divided by surface tension. And I think this is a nice result, and it comes up in many places. Okay, so what does that have to do with us? I was interested in the question of suspensions. What happens if you have particles in solution and you do this case of withdrawal of a fluid film shown on the left? This was work done with Carlos Koloscu, who's now a professor at Stony Brook in New York. And the question was, if you started with a suspension, would you end up with particles in the film? And shown here is a snapshot of a numerical simulation that shows, as a function of time, dimensionless time going from left to right, that if the film is thick enough, a particle coming towards the film may get entrained in the film. And this suggested to us that there's conditions on pulling speed, which meant capillary number, which meant film thickness, that might allow you to control particle deposition in a deposited film. Okay, so my student Estella Yu, as part of her PhD, set up an experiment to do this, and the expertise of Sepeda Koda-Perast in flow visualization and measurement was crucial. The experiment is shown here. There's a small tube where we'll inject a suspension and bubbles. There's a rectangular system around it to do index refraction matching to help with the optics. And there's a camera that can move along the tube following the bubble. And so the experiment is going to be, we'll inject fluid into the tube, and then we'll inject a bubble, and we'll try to see how the bubble moves relative to the liquid or to the suspension. And the experiment is shown here. We're interested in separation, so we start with a bidispersed suspension of 2 different sized particles. We inject a bubble, and what you might see as this is playing is that there's 2 particle sizes to your right. The bubble passes with a uniform film. The film undergoes a shape change. Particles get stuck to the interface, but only small particles end up at the back. And so there are a number of different fluid mechanical questions here that you might get interested in, whether it's particles at the interface, the shape of the film, or the fact that this movement of the bubble has achieved a separation of the particles by size. So this is now then what we think about as a continuous flow process. It involves sort of a minimal pressure drop, unlike what you'd have if you tried to push things through filters. And I'll just want to mention, go through a few slides that illustrate the mechanism, and then I want to talk about effectively the criteria for separation and show you how effective this might be. The idea is shown here in a sketch that the bubble moves relative to the wall. So this is a version of the Landau-Levitch Deryagan-Brotherton problem. And if the film thickness here denoted by B instead of H is bigger than the particle size, you might expect that the particles could get in. The fluid mechanics of this problem says there's always a separatrix that separates the fluid that can enter the film from the fluid that is pushed in front of the bubble. And there's a classic result from David Curie that extends the Landau-Levitch solution as shown in the formula in the box, that B is the film thickness here instead of H, R is the radius of the tube that the bubble sits in, the numerator is the Landau-Levitch solution, and the denominator is a correction for finite capillary numbers that David Curie and his research group discovered. I'm showing on the right again the picture. We have the numerical simulations of the entrainment process, and I've shown you a movie already of what the limits might be. It turns out when the particle here denoted by d sub p is larger than the film thickness, you can imagine it's just sterically hindered and makes it very challenging for a particle to get into the film. And so rather than studying the fluid mechanics problem, we studied this separations problem. We did a large number of experiments. And here I'm going to show the, if you like, the two main variables. Capillary number on the horizontal axis, we're going to go from a capillary number of about 1 × 10⁻² to 2 × 10⁻². And we're going to study particle size on the vertical axis. So we're going to show what happens if you just have a monodispersed suspension of 10 micron particles or 40 micron particles. So at a capillary number of about 10 × 10⁻², the film thickness is almost 30 microns. That's bigger than 10 microns. So if you look at this case and you focus on the back of the bubble shown by the red box, all you see are small particles, the 10 micron sized particles in this monodispersed suspension. If you do the same experiment with 40 micron particles and look behind the bubble, then none of the you see no particles behind the bubble. The bubble effectively pushes the entire suspension in front of it. On the other hand, if you now double the capillary number, which effectively doesn't quite double the film thickness, but gets the film thickness closer to 50 microns, then in a monodispersed suspension with shown in the bottom slide of 40 micron sized particles, if you look behind the bubble, you find some of the bigger particles. So we've done experiments with monodispersed suspensions to try to identify a separation criteria, and that's shown here. If you measure the film thickness d relative to the particle diameter dp as a function of capillary number, the black line is what you'd expect for the Landau-Levitch solution. And what we find is that whenever the film thickness is larger than the particle diameter, you can expect particles to get into the film. So we call that leakage. On the other hand, if the film is ever too thin, you just can't get particles in. And the data points are shown, the microfluidic data points are shown by the dots, and the curve is what we'll call the separation criteria. And so, it's sort of just a steric condition, but it's trying to now apply this fluid mechanical idea to separations. And then we did this then with a bidispersed suspension. So we show a graph here on the lower left, which has particle size relative to film thickness. Here I've flipped the ratio. And there are two different particle sizes. So there are large particles shown in the red on the right, and a lot of small particles shown in the red at the left. And this is before we do the experiment. And then if we look after the bubble has passed, we only find small particles in the solution. This was actually the video I showed you of a bidispersed suspension of 10 and 40 micron sized particles. I should say that a nice thing about this separation criteria is the film thickness is set by speed. So if you think about what is the dial the engineer is dialing, they're just dialing the mean speed of the flow. They're not setting anything else. And that means it's a tunable parameter as maybe solution conditions change. We tried something that's a little harder for us to look at how precise this might be. We took a suspension of 10 and 20 micron sized particles. Again, in red is the bidispersed suspension before we do the separations. And then afterwards is shown in blue. If you look very closely at the graph, you'll see a very small number of the larger particles do leak through. But overall, what I'll argue is that this is an application of this Landau-Levitch problem to a separations process in a continuous flow process with a tunable parameter, which is the film thickness, which is the sets of separation criteria. And it's tunable because it only depends on flow speed. Okay, so that's my first short story about a wonderful fluid mechanics problem if you want to look at something to study offline, and a application to separations. My second short story is diffusiophoresis. This is well, I should ask Ashish, how are we doing so far? Are people still with us?
It's very nice. I think,
Okay.
Good on time also.
Okay, so this next problem is diffusiophoresis. This is just going to be a whirlwind sort of through it. It's a problem we've worked on for about 5 years and involves a whole slew of people in my group. Okay, so I'm going to do this the following way. I'm going to give a short introduction to the phenomena. There are review articles written about it. Then I'm just going to show one thing we tried, which deals with so-called multivalent electrolytes. And then one thing we tried that's a little more practical, which says that oftentimes when you deal with real systems, you have a background of electrolyte. And I just want to show you how, some of the questions that come up in this topic. So if you've studied physical chemistry, electrolyte solutions come up in many places. Typically, a surface is charged, and so you'll find this in the biological separations process that's called electrophoresis. You'll find it if you go to civil engineering departments and study how cement is made and processed. In energy storage, like batteries and supercapacitors, you have electrolytes and charged surfaces. And finally, in the physical chemistry world, just movement of particles relative to fluids, you find this. And again, I've already introduced the language, but here I've indicated in detail that if you have a particle shown in black and charged ions shown with the sign of the charge, if there's a chemical gradient, a particle will typically move. We've actually applied it to try to think about how you might filter water. And one nice thing about this subject intellectually is that it just spans all the science and engineering disciplines. So you couldn't ask for a better topic to think about, even if you're only studying one part of it. It gives you a nice toolbox. Okay, so what you learn in textbooks, as shown here, if you have a surface where charges get embedded in the surface, either due to ionization or to ions getting trapped, the surface picks up a net charge. Since the system has to be overall charge neutral, there's a counter charge near the surface that's called the Debye screening length. And outside of that region, the system is electrically neutral with as many plus as minus charges. The region of the charge separation, the so-called Debye screening length, is shown in the formula on the right, where C0 is the bulk concentration and higher concentrations mean smaller screening lengths. These dimensions are typically measured in nanometers to tens of nanometers. So this region of charge separation, which in some sense where all the action is on a scale much smaller than what you see on macroscopic dimensions. Now, the picture I've shown you is the equilibrium picture. It says that if you have any one-to-one electrolyte, like sodium chloride, potassium chloride, lithium chloride, it would say they're all the same, just from this kind of picture of charges. The question is, what if you're out of equilibrium? And what I'll say is, when you're out of equilibrium, the choice of the salt matters. Not all one-to-one electrolytes are the same. This was recognized already 80 years ago by Daryagin and colleagues in the former Soviet Union, but is not always well appreciated by people who are trained in physical chemistry and only think about equilibrium processes. So to make this clear, I'm going to show you a nice experiment from Litter Bouquet's group. But the idea is if you have a particle and it experiences a force, it moves at some velocity v relative to the fluid. And so you always have to think about what are the forces acting on the particles. And in this case, I'm going to show you, it's a gradient of chemistry that produces the force. So the experiment done by the Bouquet group I think the work of Litter Bouquet and Armand Ashdari and colleagues is shown here. It's a microfluidic experiment. There are 3 of them. It's a co-flow of a colloid in the middle with a buffer solution outside it, shown in panel A. In panel B, they add salt to the buffer solution. And in panel C, they add salt to the colloid solution. Shown on the bottom are confocal microscope images of a cross-section at different axial positions along the flow. When there's no salt added, the confocal image is uniform along the flow direction. The colloids move very, very fast, and although they're Brownian, you can't see any roughness of the interface between the yellow region where the gold colloids are and outside. But if you put salt on the outside, the central stream widens, and if you put salt on the inside, the central stream contracts. So the particles are clearly moving relative to the flow, which is primarily axial, and you're allowed to ask where the force comes from. Well, the force comes from an electric field, and the electric field comes from a chemical gradient. I should say there's clearly a chemical gradient because you're only putting salt in part of the solution, so there's always a transverse chemical gradient in panels B and C. There's a famous formula due to Smolkowski that says for the thin double layer limit, the speed that the particle moves is proportional to the electric field, and a surface potential here with the Greek letter zeta. Epsilon is a dielectric constant of the solution, and mu is the viscosity of the fluid. So you will always move if there's an electric field, and here the electric field comes from a chemical gradient. And I'll show you this again in a second, but the formula is shown here. The principal idea, not the only idea, but the principal idea for many systems is that when you have a chemical gradient, here denoted by the gradient of the logarithm of C, you get an electric field, and the prefactor depends on the difference of diffusivities of the cation D plus to the anion D minus. And that says that not all even one-to-one electrolytes are the same because D plus can be different than D minus depending on the size of the ion. And this also says that although you'll read theory papers that say the driving force for motion for V, which is proportional to E, you might guess would go like Fick's law as the gradient of C. That's not true. It varies as the gradient of the logarithm of C. And so there's an additional chemical dependence. Okay, so this is just saying chemical gradients create local electric fields. Those cause particles to move. To give you some idea of what this means, I've shown you the diffusion constants for sodium chloride and potassium chloride. You'll see that the K plus and the Cl minus have nearly identical diffusion coefficients. So therefore, diffusiophoresis is very weak in KCl, but there's a 50% difference between Na plus and Cl minus, if you can see my cursor. And so diffusiophoresis is quite strong in NaCl. And therefore, you can expect that the choice of salt now matters in this kind of problem. And if you only calculate the change in the electric potential, that's just shown here. Its gradient gives you the electric field. And in the physical chemistry literature, it's been known for decades. This kind of idea has been known for decades. And there, the language is, this is called the diffusion potential, because it's the diffusion of ions that give rise to changes in electric potential. And so if you want to think about this problem that I just showed you, why do the particles move relative to the fluid? It's because of the chemical gradient. And there are many, many studies now on this. And in recent years, I've listed many of the wonderful groups that have been exploring these kind of questions. Okay, so we were interested in different kinds of flows, maybe mimicked by porous media. So we used microfluidic methods to look at so-called dead-end pores. Work by Darryl Velogle's group at Penn State a little before ours had also been studying this problem. I've outlined the dead-end pore here. There's a colloid that's going to flow left to right along the top as indicated by the image. And if you play the movie, what you'll see is that the colloids are driven into the pore. The pore is a dead-end pore, so there's no flow into it. It's just the particles can move into it because there's a chemical concentration difference between the pore and the mainstream. That's a chemical gradient that drives the particles. And We used two different particles here of different sizes, and you'll see that the large particles here, fluorescently labeled in green, move a little faster than the red particles. And so this also says that you can move particles into a dead-end pore. You can also then move them out of a pore. We've done this, as have several other groups, with different kinds of particles, whether it's charged macromolecules, droplets, vesicles, or cells. We also do the experiment a different way. Rather than having the particles start outside the pore, we can often start them inside the pore. That's shown here on the right, where we fill a channel with particles. We then flow an air bubble past it to just leave particles in the pore, and then we can change the solution outside the pore to see what happens. And we call these kind of experiments, when you do them, compaction experiments, because the particles are all starting in the pore. In some sense, it's a little cleaner microfluidic experiment to do image processing on. And in that experiment, when you have a gradient of chemical, here again, NaCl between the outside and the inside, the particles are compressed, and because we have many identical channels, we get many experiments all at the same time. So that's roughly diffusiophoresis due to a chemical gradient. Now, everything I've shown you and everything in the literature is on one-to-one electrolytes like NaCl or KCl. So we got interested in what happens when you weren't a one-to-one electrolyte. And so we used microfluidics as a toolbox for studying a wide range of different so-called ZZ electrolytes. So we did 2-1 electrolytes or 1-2 electrolytes, et cetera. And we compared with theory, which I haven't shown you, but we compared with theory to just try to understand how well the theories, which are necessarily approximate, do in predicting the results. A typical experiment, when you have particles moving into the pore, is shown on the top. You start with no particles in the pore, you apply the chemical gradient, and particles move into the pore. You can track the light intensity as a function of position in the pore, and do this with whatever salt you want. Here we chose a 1-2 electrolyte, Na plus and SO4 2 minus. We also did our compaction experiment, since it's an experiment my postdoc Sue and Shim developed, and it allows a lot of also further control. And that also allows us to measure light intensity in the pore as a function of position. If you compare with theory, this is a slide just showing you that there's a reasonable agreement, and it shows an effect of the salt. So the choice of the ion matters. And we've shown it here for 3 different salts on the left, middle, and right. And we track position of the front versus time. The dots are all the experiments. The curves are the prediction of the theory. In both cases, it's not perfect agreement, but it's not bad. And in both cases, it shows the difference of the electrolyte. And in all cases, it shows that the chloride salt is more effective than sulfate anion. So again, it's telling you something about the choice of the salt and the details of the physical chemistry acting on the hydrodynamics. And so there's things that you can then learn in detail, which we find quite interesting when we're trying to think more about how physical chemistry impacts problems in engineering and biology. So that was 2 slides showing that. And then another question we asked about, in part because we're sometimes thinking about cleaning water, and I should say I'm part of a little startup thinking about these questions, is what's the role of a background electrolyte? You know, if you do an experiment in the lab, you can control everything, but if you get some fluid from someone, it could have all kinds of salts in them. And in fact, in the literature, there's sort of ad hoc models, which, while plausible, are sort of suspect. And they go like this. If you have 2 electrolytes here labeled A and B, and I've labeled them with a common cation shown in purple, but a different anion shown in orange and green. When you mix these 2 salts together, one set of modeling in the literature, which is plausible and sort of chosen for simplicity, is you always think about the two salts as independent. So you think about just mixing two different things and you treat them each as their own electrolyte. On the other hand, and I'll call that the independent electrolyte situation in the graph on the right, which I'll explain later, but that's, you could sort of always treat NaCl and KCl as separate, or you could say, well, there's always Brownian motion, they're always mixed up. They're really three coupled ions together. They're not two coupled salts. And so when you have three coupled ions together, we'll call that the coupled theory. And that's what we've tried to explore, which we'll call the real solution. And we studied this experimentally using a system that had three ions in it. So again, we chose a common cation. We did that using a fluorescently labeled sodium salt called sodium fluorescein, and then other salts. And so the sort of experiment is shown on the right. There's a fluorescent salt in the channel, and we're flowing in, letting another salt invade into it. So this is a problem with three ions, but we can track one of them because the sodium salt is fluorescent. Okay, so that's the kind of experiment we do. And the moral of this story is that the gradient of one electrolyte drives the rearrangement of other ions in the solution. And I've shown you the movie in the middle already. And so you can plot intensity versus position in this experiment for different times. And then we can also do experiments with different kinds of salts. And then we can also do theory. And the theory is shown here, just in sketch. You have three different ions. So there's three different choices of I, 1, 2, and 3. Here we have two anions and one cation. The diffusion equation, if you like, has both a normal diffusion term with the diffusivity di, and it has an electromigration term, which is the last term. The electromigration term involves the electric potential psi, and the electric field comes from Gauss's law, requires that you know the distribution of all the ions. We'll call this the coupled theory. It's in the past, as I said, people had mostly just forgotten the electric field and just treated this as diffusion of two salts. And this isn't the clearest slide, but we did lots of experiments and we compare the experimental results here shown by the dotted curve with the coupled model, which is shown by the solid curve. And in all cases, which we describe in our paper, the coupled model did a little better than the approximate model, which is treating it as two separate salts. And again, this is showing concentration versus position in one of these compaction experiments as a function of for different times. So it's a bit cluttered, but in all cases, you can try to find that the experiment is always a little faster than the model, but the model tracks the approximate time scale of what goes on. And the model effectively has no fitting parameters. It's just taking diffusion coefficients from the literature. And as I said, the coupled model agrees better than the model that treats these electrolytes as completely uncoupled, which is what's typically described in a mass transfer book. Okay. Ashi, I think I've used my 45 minutes. I have a short story left, but I'm happy just to skip it and handle questions. I don't want to completely bore people, but it's really up to you.
We are fine to, listen to another maybe 10, 15 minutes. If
well, do you have any, are there any questions on the first two parts before I go on?
There are some questions in the chat box. I see them already. So
would you like me to handle them now?
We could do that probably at the end if you agree.
Okay, then this is short. Let me see if I can do this in 10 minutes, but hopefully this has been clear. Okay, so this has to do with a question that, if you pay attention to the climate problem, involves carbon, and I'll refer to this as the soil carbon problem. Okay, if you follow the news, you know that more and more rapidly, we are learning that it not only appears that there's climate change or modification, but there are certainly indications from the IPCC that it's happening more and more rapidly. And in this report, which was published 3 weeks ago, it emphasizes the need for immediate, rapid, and large-scale reductions in greenhouse gases. And one way people sometimes think about is to recognize that although the atmosphere has a lot of carbon, shown in the circle on the right, soil has 5 times more carbon typically stored in it. So this has both pluses and minuses. And I should say, this is a joint effort with my former postdoc, Judy Yang, who's now at the University of Minnesota, but 2 Princeton colleagues. And I should say, Ian Borg is an expert on this topic, and we heard a talk on this topic. And after listening to the talk, which highlighted all the challenges and many of the assumptions in modeling, Ian, Jining, and I, and colleague Milcare Porpado at Princeton asked, aren't there experimental tools we can use to test some of the assumptions that people make in models? And the microfluidic study I'll show you is an attempt at that. Now, in a recent report from the National Academy of Sciences in the United States of America, they note that there's the possibility of what you might call a negative emission technology, which is, could soils provide a reservoir to store carbon? So that would be a good thing. On the other hand, they do note that a warming climate might release more of the carbon from the soil. That would even be worse. So there are pluses and minuses. And if you ask what are predictions from soil carbon models, on this slide, there's the amount of carbon in soil on the vertical axis, there's the year on the horizontal axis. The results from 3 different models are shown. I'm no expert on the models, but they make different kinds of assumptions. You can see that the predictions are all over the map. There's huge variability. And so you might think that because there's so much carbon there, and you might either be able to store it or lose it, it would be important to understand different assumptions in models to make models better. And that's one thing I really like, I think many of us like, about science and engineering, and microfluidics provides us a toolkit. In this problem, clay is one of the so-called key carbon capture materials for reasons I'll tell you about. And I'll just point out, I added this slide for you to just point out that if you go to the literature, there are even modern puzzles, observations people have made without any explanation. The link between carbon and clay goes like this. Organic carbon is often considered to be adsorbed to clay, and clay can form aggregates, which can trap the carbon shown here in orange. The carbon storage feature is built on a hypothesis that clay can protect carbon, and they can protect carbon, it's thought, because bacteria can't get in to feed on the carbon. Okay, so that's thought. On the other hand, there are some experiments that have noted that whenever you add sugars to solutions, it also often leads to release of carbon that's trapped in the soil, and that's called the priming effect, though there's no explanation for that. Okay, so with that, I'll now go forward. So I was aware when I heard a nice talk on the soil carbon problem, I was aware of a paper from a UK group and they call it Soil on a Chip. And there's beautiful work also from Toby Kears and Tom Chamizo and colleagues in Amsterdam on this idea as well. And so we're going to use the soil on a chip idea. We're going to take a clay, which is transparent, called lapunite. It's similar to other smectite clays. It's transparent so you can look into it. We're going to use different kinds of organic molecules that are fluorescently labeled. And we're going to do confocal microscopy in a microfluidic setup to allow us to flush in water, look at organic materials, and just see what happens over time. And when we have to do experiments that are biologically relevant or environmentally relevant, we're going to add a soil bacterium, in this case also Pseudomonas Originosa. So these are all the elements that go into our paper, which appeared only earlier this year. The typical experiment we show here is a small molecule carbon, just glucose. You see playing on the left a confocal microscope image of a clay aggregate. We deposit a solution containing glucose, then we flush it with fresh water. And you'll see that although glucose can first get trapped inside the clay, when you flush it with fresh water starting at around 2 hours, all the glucose is removed. So these are the kinds of experiments we try to do to look at what happens when you have clay and carbon in water, like would happen in soil. If we do the same thing, though, with a large molecular weight dextran, just here, say 3,000 to 5,000 molecular weight, but we've gone higher, then once you deposit the carbon, it gets trapped in the clay aggregate, and then it typically stays for many tens of hours, and we refer to that as irreversible absorption, or at least, approximately irreversible over the course of several days. So these are the kind of experiments we do to probe carbon and clay in a microfluidic setup that mimics a soil. Now, the question is, what happens when you have bacteria? And when you have bacteria shown here in red with the green fluorescently labeled carbon trapped in the clay, the bacteria can't get inside the clay aggregate. And so that's fitting with the a hypothesis I told you earlier, that the bacteria the carbon is protected by the clay aggregate because the bacteria, which are micron size, can't get into the nanoscale pores in the soil. So you might think that's good, but bacteria can produce enzymes. And so we did experiments by adding extra enzyme. We refer to that as exogenous addition of an enzyme. I'm going to play a movie that shows, playing in the upper left corner, you'll see the kind of materials in the stream. And then we'll first deposit different molecular weight carbons shown in color, and then we'll add an enzyme. And so now when you do a microfluidic experiment with the soil aggregates at the bottom, the carbon gets trapped inside or on the surface of the soil. But when you add an enzyme here called dextranase, it rapidly diffuses, gets into the clay aggregates and removes all the carbon. I'll play it again. You deposit carbon for small molecular weight and large molecular weight. Then you flush with water, and now you flush with water, and then you add enzyme. And the enzyme effectively gets into the pores and removes all the carbon. And so this is suggestive that the bacteria do have an effect on the system, and it rationalizes priming, because sugar can cause bacteria to grow. When bacteria grow, they produce enzymes, and enzymes then can sneak their way into clay aggregates and release, act on molecules and release the carbon molecules. And so if you go to the literature and you study this part of environmental science, a typical picture for the absorption of low molecular weight molecules, which is called reversible absorption, or high molecular weight molecules, which is called irreversible absorption, is shown here. The models typically don't have biology, which is bacteria. And so our suggestion is that, in fact, the bacteria are producing enzymes that are breaking down large molecular weight materials, causing them to become low molecular weight materials, and therefore they're really, in the presence of biology, going to be reversibly absorbed, which in some sense is a long-term problem and makes carbon storage challenging unless you can control the environment more carefully. But this overall says that these kind of climate models trying to address soil questions need better experiments, and that's a role for people like those on the call and myself and colleagues. Okay, so that's my talk. I've shown you different experiments from fluid mechanics to diffusiophoresis to bubble separation to separation processes of bubbles to this quick sketch of a soil carbon problem. What they have in common, at least the last three, are the use of microfluidics to try to ask new questions. And with that, I realized I've used 55 minutes, so I'll stop and happy to answer questions. And thank you for giving me the chance to talk to you from halfway around the world. But I guess it's one feature of Zoom that means that we can still be colleagues when traveling is difficult.
Thank you, Professor Stone, for a very motivating talk. And I already see a few questions in the chat box, which I would moderate. And then after that, I have some of my colleagues who have joined. I have made them co-hosts, so they can probably unmute and ask any questions that they may have. So I see the first question is about
yeah, I can see them, so I can try to handle them. The first one I see is, what is the length scale associated with colonization? Is that what you see?
Yeah, yeah.
Yeah, so in that experiment I just showed, it's a bacteria that it sticks to a surface. It has a cell that grows another cell, and so the length scale is effectively on the order of 2 to 3 particle cell lengths. It's always colonizing locally, unless it breaks free and then it gets completely swept away. So it's a completely local colonization problem. And the suggestion we made was that the benefit to the bacteria of a curved shape is you're able to colonize. If you're a straight shape, you grow like this. You're stuck to a surface, the next one grows, it's over here. When it breaks free, it's caught by the flow and swept away. You're never able then to colonize locally.
I had actually a related question. Maybe I could ask that before we move on to another question. So you showed about this upstream swimming of bacteria. And we also see the same in other active matter like sperm cells and C. elegans and other systems. So is there a flow rate limit where such an upstream transport?
Okay, so I think if you're referring to upstream migration of E. coli,
yeah,
that's a swimming cell. So the active matter are swimming cells, and the upstream migration is a hydrodynamic effect of the interaction of the swimming cell with the wall. In the case I showed you, the cells are on the wall. They're physically stuck on the wall. When the cell attaches to the wall, it typically loses its flagella, but it has these pili that allow it to pull. So it has surface motility. And so it migrates upstream, but for, I think, a very different reason than the upstream swimming cells that you might think about in the active matter world. It's still active, but it's a different kind of upstream movement.
Is that because of the chemical gradient, because of some secretion?
Why the movie I showed you goes upstream?
Yeah.
No, I tried to show a quick picture, but it went fast. So here's a cell. And on one end, it's going to have these so-called type 4 pili. So when the cell gets stuck to the surface, it sticks with the type 4 pili. If now it sees a shear flow, it's going to get tilted. And now, no matter which orientation in the plane it gets tilted, the pili that are holding it, it can then move, are all directed upstream. And so if it actuates the pili, it's going to have an upstream component to it. Okay. So in some sense, the flow is orienting the surface attached cells.
Thank you. The next question is about leakage. I think this is about the bubble problem.
Yeah, so to a first approximation, we view that, let's say I have to think about, we would argue that a sufficient condition in most cases is that the particle diameter being smaller than the film thickness would be a condition for leakage, a simply steric condition. And the film thickness is set by the mean speed, by this Bretherton or Landau-Levitch problem. So that you select by hydrodynamics, and then you get this condition. Yeah. I know there are a few other papers that have a prefactor that's not quite one, but in our experiments, it was always.
But will concentration also play a role? Because I think
we only studied dilute systems. We only studied dilute systems. So if I had to make an estimate, I would say concentration affects the mean viscosity. The mean viscosity sets the film thickness.
Yeah.
In the experiment with diffusiophoresis, bigger particles move faster. That's outside the limit, the electrokinetic limit of the Shmolkowski result. But it is a feature that's predicted by more detailed theory from John Anderson and Dennis Prevey. And in our paper, we suggest that if you use their more general theory, then you get this kind of result. What is the herringbone pattern?
Yeah, I think probably, this was a general question, and I had asked, who had asked to ask question related to the talk. Probably that's unrelated to the talk.
Oh, are you asking a question about the herringbone?
Yeah.
It's not something I showed, but is that something you're
Yeah, I
asking
think
about?
probably you can skip that question. I think the
Well,
question.
I have a slide I can show if it's helpful,
channel.
but in another talk. But it's the herringbone pattern is if you have an axial flow, it helps create a transverse flow. And so that transverse flow can help with different kinds of mixing. So the herringbone pattern is used in microfluidics, I think, typically when you're thinking about trying to do things that you might call passive mixing. There's a flow, but you want to create some kind of transverse convection. It's not strong, but it is in some sense always there. Can we expect a better separation if the droplet is intermittently actuated instead of constant flow? I don't. So there's a question. Can we expect better separation of the droplet that entrains particles is intermittently actuated instead of constant flow? I
think it's the bubble that he's talking about.
Yeah, no, I understand. I don't think so. It's not something we looked at specifically in the separations problem. These low Reynolds number problems typically adjust reasonably quickly the flow field. And so I don't think it would have a big impact on separations. On the other hand, you might have noticed in the bubble problem that there was a thin film upstream and a thicker film downstream. And so the film thickness is speed dependent. So if you have a bubble moving at one speed with a thin film, and now you change the speed, the film thickness has to change. And that transient is hydrodynamically interesting to study. And we did write a paper about how that transient in film thickness develops. But I don't think it has a big impact on separations. Single cell genomics, there's a huge literature, I think now, on droplet microfluidics and single cell biology, whether it's bacterial growth, chemical effects, drug discovery, et cetera, because the droplet can be an isolated chemical reactor. So there are startup companies and lots of labs around the world doing droplet microfluidics specifically for different questions in cell biology, from genomics to proteomics to your favorite omics. In case of polydisperse particles in a fluid medium, in case large particles disperse, can they affect the entrainment? It's a great question. I think that relates to Professor Sen's question about concentrations. We only looked at relatively dilute systems, and we didn't really notice anything about the interactions of the two particles, but it is an interesting question whether the big particles would tend to block the smaller particles from somehow entering the pore. That's a great question. We've published our 1 or 2 papers on it, and I've moved on. But I do think it's an interesting area to ask about, if you like, membrane-free filtration, because the bubbles here act as sort of the speed-dependent filters. Yeah, and I don't have any methods to avoid such problems if they were to happen, at least none off the top of my head. It's a great question. Okay, we did all the questions. Ashish.
Some colleagues, if you have any question, please try to unmute and you can ask your question to Professor Stone. This carbon work was very interesting.
I'm sorry, I couldn't hear that.
Your work on carbon.
Yeah, I will. That it really happened the way I said. I heard a talk about 3 years ago, and it was a great talk about how to think about this question and the different assumptions people make. And at the end of the talk, I simply asked, why doesn't someone check some of the assumptions? It has carbon and soil particles and clay and water, and bacteria. And Judy Yang joined my group and took up the challenge. And I'm sure there are other people that can do, more things like this. But I really found it interesting that I asked all these experts, why doesn't someone check the assumptions? And the answer was no one had, or at least that's how I understood it. And we had a very nice collaboration with several of my colleagues, Yin Zhang and Ian Borg, who come from different fields, to try to ask how a microfluidic toolbox might give us some insight.
Yeah,
and it points out it's complicated.
Handling clay inside microfluidic channels is not easy.
Handling what?
Handling clay. I was saying inside microfluidic channels could lead to clogging and other issues. So, it's very nice what has been done.
Yep, Professor Stone, this is follow-up. So in the last problem of clay, is the clay moving on the channel or the clay is stationary?
So normally what we had were just little clay aggregates that got stuck in the channel. And for us, that was fine because it looked sort of like an aggregate in a soil. And so we just worked with what we had based on that.
But how the bacterial loading changes the carbon dioxide storage on the clay? Like if you change the bacteria.
Not carbon dioxide, but carbon storage. So what we found was if we just used the bacteria, they would get stuck on the outside of the aggregate and they weren't able to access the inside of the clay aggregate. That was sort of a hypothesis that people had made in literature. If we add an enzyme that bacteria can produce, it can get inside the clay and degrade carbon, and then the carbon gets released. The one experiment we weren't able to do, all of this took place really during the pandemic where there were all kinds of challenges, was to do an experiment with a bacteria that was actively producing the enzyme. So that experiment we didn't do. But we did the two pieces of it. We showed that the bacteria are stuck outside the aggregate, but if there was an enzyme present, it can get inside the aggregate where carbon might be trapped. If the enzyme breaks down the carbon and becomes small molecules, I showed you already movies that those are released. So we sort of did all the steps that we think might be taking place, except for one experiment, which is bacterium actively producing an enzyme.
Very nice work.
You're in charge, Ashi.
Yeah, it looks like no more questions. So let's thank Professor Stone again. And
you've tired me out, but it's only 8 o'clock in the morning for me, so I have my whole day in front of me.
So we hope that we'll host you sometime at IIT Madras when the travel resumes. And
that would be very nice.
So thank you again for the talk.
Okay.
And, have a good day.
All right. If there's nothing else, then I'll sign off.
Yeah. Bye-bye.
Okay. Have a great day. Thank you for the opportunity.